Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on digestion of proteins. Starting with the first point, the title, the digestion of proteins. Now, first question, where the process of digestion is taking place in our body? So, the process of digestion begins in the stomach. Digestion of protein starts in the stomach with the action of pepsin. So first of all, the pepsin acts on protein in the stomach, and this digestion which has begun in the stomach, this process is completed in small intestine, and the enzymes which are involved in the completion of protein digestion, they are present in the pancreatic juice, and also they are located on the breast border of enterocyte cell. So here the enzymes are present in the small intestine, the pancreatic juice, and the breast border. Proteases. Now there are two classes of proteases, that are the endopeptidases and the exopeptidases. Now the endopeptidases, they hydrolyze the interior peptide bonds of the protein. The role the endopeptidases are playing, that they are hydrolyzing the interior peptide bond of the protein. Now the endopeptidases of GI tract are pepsin, trypsin, chymotrypsin and elastase. These are the endopeptidases of GI tract. So this was the first point, where the process of digestion is beginning, where it is completing, and which are the enzymes that are involved in the process of digestion. Now moving on the second point. Now the exopeptidase. Carboxypeptidase A and carboxypeptidase B, these are the two exopeptidase. And what is the function of this carboxypeptidase A and B? So they are hydrolyzing one amino acid from where? So C-terminal end of the protein. C-terminal means carboxylic acid terminal, COOH group. So from that end, they are removing one amino acid from the protein. So that is the role these two enzymes are playing. And if you remember, I have already delivered the lecture on the pancreatic juice, and in that lecture in detail I have explained the mechanism in which they are working and they are removing this amino acid. So now we are moving on the third point. Now protein digestion, as we know, it has started in the stomach, and this protein digestion continues in the small intestine. Once the digestion of protein completes in the stomach, we know the chyme is formed, and then the chyme enters into the duodenum, the first part of the small intestine. So the process of digestion is continuing in the small intestine, and in the intestine, on the protein, the pancreatic enzymes, they are involved in the protein digestion, and in the small intestine, the breast border proteases are also involved in protein digestion. Now the 5 major pancreatic proteases, and these proteases are actually they are in present in an inactive form. So they are the inactive precursors of enzyme. So their name are trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A, and procarboxypeptidase B. Third point. Now here I am showing you one figure. So this is a very good figure, and here you will get the idea that in the stomach what is happening. So see, in the stomach, pepsinogen and the acidic pH, or due to the HCl, the pepsinogen which was in inactive form, so now it is converted into active form by the action of HCl. So now this pepsin is in the active form, and this pepsin now it is ready to participate in the protein digestion in the stomach. Now fourth point. Now what happens in the intestinal protein digestion? So the first step in intestinal protein digestion is the activation of trypsinogen. Trypsinogen is we can say it is in the inactive form, so we have to convert it into active form, and the active form is the trypsin. Now the enzyme which is responsible for the activation of trypsinogen and conversion of trypsinogen into trypsin, that enzyme is enterokinase. And where this enterokinase enzyme is located, so this enzyme is present on the brush border of the enterocyte. So here you see in the figure that this is the small intestine we are talking, trypsinogen, then the brush border enzyme enterokinase, and this enterokinase working on trypsinogen, and it is activating trypsinogen into active form trypsin. So the fourth point. Now moving on the fifth point of our discussion. Now here the entire theory point is written. You can go through this theory point, but I am directly explaining this theory point. So here in the figure you can see trypsinogen is activated, so we are having active trypsin. Now you see what is happening on trypsinogen. Trypsin is acting, so trypsinogen is converted into active form, that is trypsin. Now this trypsin acts on inactive chymotrypsinogen, so chymotrypsinogen is converted into chymotrypsin. Trypsin act on proelastase, so proelastase is converted into elastase. Trypsin act on procarboxypeptidase A, so procarboxypeptidase A is converted into carboxypeptidase A. Trypsin act on procarboxypeptidase B, so procarboxypeptidase B converted into active carboxypeptidase B. This is the fifth point. Now moving on the sixth point. Now here you see protein digestion in the stomach, lumen of stomach, protein, pepsin is acting on protein, so amino acid and oligopeptides are formed. Now in the small intestine what is happening? So small intestine. Now in the small intestine, in the pancreatic juice, Pancreatic proteases, they hydrolyze dietary protein. The protein of our food are known as the dietary protein. So they are hydrolyzing the dietary protein to, and they are converting this dietary protein into amino acid, dipeptide, tripeptide, and larger peptides. And these larger peptides are known as oligopeptide. So once again, pancreatic proteases in the small intestine, they are hydrolyzing the dietary protein into amino acid, dipeptide, tripeptide, and the larger peptide. And larger peptides are known as the oligopeptides. So here in the small intestine, protein is acted upon by trypsin, chymotrypsin, elastase, carboxypeptidase A and carboxypeptidase B in the lumen of small intestine. And what is happening? Protein is converted into amino acid, dipeptide, tripeptide. Then the larger peptides are known as the oligopeptide. And these oligopeptides, they are acted upon by peptidase. And where these peptidase are located, they are present on the breast border. And this peptidase, they are acting on oligopeptide. So oligopeptide, they are converted into amino acid, then dipeptide, and the tripeptides are formed from the oligopeptide digestion. So I am showing you this entire figure to you. This is also a good one. So this way. Now moving on the 7th point. This is the 7th point. Now only the amino acid, only the amino acid, dipeptide and tripeptide are absorbable. That means, let's say I am drawing, this is the enterocyte cell, this is the adjacent enterocyte cell, okay, here this is enterocyte, okay. So in the enterocyte, amino acid can enter, dipeptide can enter, tripeptide can enter. This entry of amino acid, dipeptide and tripeptide in the enterocyte is known as the process of absorption, and it is the first part of absorption process. And when it is completed, so when they are entering into blood capillary, then we can say the absorption process of amino acid is completed. Now the oligopeptide, they are further hydrolyzed by breast border proteases, and once they are further hydrolyzed, these oligopeptides, so they produce smaller absorbable molecules. And once the function of pancreatic protease is completed, they digest themselves, and at the same time they digest the other proteases. So that means they digest themselves and they digest each other when their function is over. Now moving on the 8th point. Now in the 8th point, we are talking about breast border. What is breast border? First of all, this is the epithelium, that means the enterocyte cell of the small intestine. This one is also the enterocyte cell of small intestine. This one enterocyte cell of small intestine, and this green color part is the cytoplasm of enterocyte. Then here also this is the enterocyte, epithelial cell of small intestine. Now here you see these are the microvilli. These are all microvilli, and together this microvilli, they look like the bristle of the brush, and that's why these all microvilli together, they known as the brush border. And as you can see on the brush border, this is the enzyme, this one is the enzyme, this is the enzyme, then here this is the enzyme. So on the brush border the enzymes are located. And what are the names of this enzyme? So these enzymes are the peptidases, and the name of this peptidases are aminopeptidases, dipeptidases, tripeptidases, and nuclease. And related enzymes are present on brush border. Now, what these enzymes are doing? What is the role they are playing? So this enzyme, they continue the digestive process which has begun by the pancreatic proteases. So once the pancreatic proteases digest the protein, then protein is further digested by the brush border enzyme. Okay, and this brush border enzyme, this peptidases, they convert the protein into small peptide and the amino acids. So here very good conceptual chart, this one. This chart or this concept map or this figure is very important for you, and I am explaining this figure, okay, what this figure is all about. So here you see this one is the lumen. 1 minute. This is the lumen of small intestine, and protein digestion starts in the stomach with the pepsin, that is gastric peptidase, and in the lumen of small intestine, pancreatic juices, that means the proteases, and the proteases are pancreatic peptidases, because the proteases, they are having two class, exopeptidase and endopeptidase. So these peptidases are actually proteases. So now what is happening? How the protein is digested? So on the protein, pepsin is acting, then this proteases, pancreatic, okay, they are also, and we know there are 5 proteases, trypsin, chymotrypsin, etc. All this, they are acting on this protein. So now protein is digested, and from the protein there is a formation of oligopeptide. And this what is this oligopeptide? So oligopeptide are the larger peptides, and then the removal of amino acid. So protein is digested, oligopeptides are formed, and amino acids are released. And where these amino acids are going? So here the amino acid is shown. This is the brush border and this amino acid, they are transported by carrier protein or by transporter, and they are entering into the cytoplasm of the enterocyte cell. amino acid are entering. Now what happened to this oligopeptidase? What happened to this oligopeptides? So you see the formula AAN. AA means amino acid and N means many amino acid joined together by mainly the peptide bond, and you know the structure of protein, so some other bonds are also present in the structure of protein, but the main bonds are the peptide bonds. So we can say many amino acid they are joined together by various kind of bonds. So here now this oligopeptide, where it is going, so here you see this is the oligopeptide. On this oligopeptide is acted upon by brush border peptidase. And which are the brush border peptidase? So now we are knowing the name: aminopeptidase, dipeptidase, tripeptidase, nuclease, and some other related enzyme. So these are the brush border peptidase, and what they are doing, so this oligopeptidase, this goes on the surface of peptidase. So now oligopeptidase is converted into AA4. What is the meaning of AA4? 4 amino acid, they are joined by peptide bond. So that means AA4 is the tetrapeptide. Okay, now what happened to this tetrapeptide? So this tetrapeptide attaches, or this tetrapeptide is acted upon by the brush border peptidase. So AA4 is converted into AA3 plus AA, and AA, here the AA is shown, amino acid, and this can enter into the cytoplasm by the carrier protein. Then see this AA3. Now for this AA3 means amino acid, amino acid, amino acid, that means tripeptide. So here once the tripeptide is formed, there are two routes for this tripeptide. This tripeptide can enter into the cytoplasm, or it can further digested. Okay, so now here with this red arrow, here the tripeptide is entering in the cytoplasm, or this tripeptide you see is moving forward and it is acted by brush border peptidase. So AA3 is converted into AA2 plus we can write AA. So AA2 means dipeptide is formed plus amino acid is formed. Now AA can enter into cytoplasm. And what happens to this AA2? Now this AA2, there are two routes for AA2. AA2, that means dipeptide can enter into the cytoplasm, or AA2 can be further digested by peptidase. So dipeptide acted upon by peptidase. So the peptide bond is broken down and we are having AA plus AA, amino acid plus amino acid, and this amino acid, they are entering into the cytoplasm. So in the cytoplasm, what is there? In the cytoplasm we are having AA3 tripeptide, we are having AA2, that means dipeptide, and we are having AA, that means amino acid we are having. So 9th point here. Now this 9th point, intracellular peptidases. Enzymes are present in the cytoplasm of enterocyte, and the enzyme which are present over here, this is the cytoplasm, green color, and in this green color cytoplasm, this is the color they are used, not the green color, but I am telling you here the cytoplasm is shown by green color. So in the cytoplasm, enzymes are present, and these enzymes are intracellular. Intra means within the cell. Peptidase are present. And this enzyme, intracellular peptidase, are the proteolytic enzyme. And this enzyme are present, as I said earlier, that they are present in the cytoplasm or the cytosol of epithelial cell, that are the enterocyte cell of small intestine. And what this peptidase, the enzyme which are present in the cytosol, what they are doing? So they are specific for the linkage between the various amino acid. So they are acting on particular bond between the amino acid within the minute. And this intracellular peptidases, they are digesting the dipeptide and the tripeptide, and they are converting this dipeptide and tripeptide into amino acid. So in the figure I am showing you, let's say this is the, as you can see in the cytoplasm, this tripeptide has entered, and on this tripeptide, this tripeptidase enzyme is acting. So 1, 2, 3 amino acid are formed in the cytoplasm, and over here AA2. So on this dipeptidase is acting, and amino acid are formed. Okay, and now where this AA3, so AA3, one root is the amino acid, then this arrow, what is the meaning of this? Then AA2, they are digested into AA plus AA, amino acid plus amino, or this arrow. So for that I will deliver the separate lecture on the process of absorption of protein. So to explain that, I have to deliver one more lecture. So then you will understand why these arrows are shown. So here, how in the cytoplasm of small intestine, the protein, which is in the form of tripeptide and dipeptide, is further digested and it is converted into amino acid. Okay, so now the last point, end product of protein digestion. What we are getting once the proteins are completely digested. So protein digestion has started in the stomach, and the digestion of protein completes in the enterocyte of the small intestine, and the end product of protein digestion that we are getting are amino acids. Now before I wind up this presentation lecture, I would like to show you this figure completely, because this one is the very important figure. So you go through this entire figure, and this way, and all the information, I request you read thoroughly this information, because this information is very important for you to understand the protein digestion. That's why. So with this, I have completed the presentation lecture on digestion of protein. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.